I'd like to thank you for taking the time today to learn a bit more about the IonFlux automated patch clamp system. A few process details before we get started. As Mike noted, there is a WebEx Q&A panel for submitting questions during the course of the presentation. We have a rather large audience today, but we will get to as many of the questions as possible after the formal presentation, which will run approximately 40 minutes. If we don't get to all of the questions, we will respond via email. Getting started then. Our goal in designing the IonFlux system was to raise the bar in terms of performance of automated patch clamp, as well as lower the cost. We've made significant progress in achieving these design imperatives with the systems that we offer. In terms of raising the bar for performance, our microfluidic design allows for continuous flow during the experiment and exceptional fluid exchange times. As well, we are able to incorporate temperature control, allowing for automated experiments that can be run at physiological temperature. With regard to lowering the price, we have hit an important milestone with the IonFlux 16 system, which sells for a price comparable to a manual patch clamp rig, making automated patch clamp accessible to researchers who have the need for higher throughput electrophysiology. Today's talk is geared primarily toward the research audience. For those of you who are not familiar with Fluxion, we are a privately held, venture-backed company that was founded in 2005 out of technology developed at the University of California, Berkeley's Sensor and Actuator Center. The company currently has 30 employees with 4 products commercialized and selling globally. We've been awarded 5 NIH Small Business Research Grants, including Phase 2 development grants. As well, we have been a 2-time R&D 100 award winner, winning awards for both the BioFlux and IonFlux platforms. We have 2 product lines, the BioFlux systems, which are used for studying the impact of flow and cell-cell interaction. As well, we have the subject of today's webinar, the IonFlux system for automated patch clamp, which includes the IonFlux 16 and IonFlux HT. Shown on this slide is a partial list of our customer base for both the BioFlux and IonFlux platforms. Fluxion has a good track record of delivering innovative products to the marketplace and offers industry-leading customer support services. Simply put, the IonFlux 16 system offers the capability to dramatically increase your lab's capacity for obtaining electrophysiology data quickly and reliably for roughly the price of an additional patch clamp rig. This is achieved by running experiments in parallel, up to 16 experiments in the case of an IonFlux 16, or 64 experiments in the case of an IonFlux HT. Additionally, the training time required to get your lab personnel up to data acquisition speed is greatly reduced due to the automated patching process. This allows for greater consistency in the data being produced. Finally, the space required to achieve this is a small fraction of that occupied by an additional patch clamp rig. The key applications for the IonFlux system are those where recombinant cell lines can be used and where a large data set is desired. Chief amongst these types of activity is compound profiling, the key application that has been driving the adoption of patch clamp automation in the pharmaceutical industry for the better part of the past decade. With an IonFlux 16 system, one could screen up to 64 compounds at a single concentration or produce 8-point IC50 curves in a single run, and 4 times this with an IonFlux HT system. Alternatively, the system could be used for screening clones, allowing up to 8 clones, or 32 for an HT system, to be compared in parallel. Additionally, one could use this model for comparing the impact of mutations on ion channel expression and function. Taking a closer look at the benchmark, the manual patch clamp experiment, shown is a typical example of a manual patch clamp rig consisting of a microscope for visualizing the cell to be patched, manipulators for positioning the patch, pipette, and perfusion lines, and the perfusion system made from syringes, all of this contained within a Faraday cage. Not shown here are the patch clamp amplifier, digitizer, data acquisition computer, and accessories like a bath temperature controller, an oscilloscope, signal conditioning hardware, or valve controllers. The great advantage of the manual approach is the overall flexibility, allowing either adherent or suspension cells to be used, as well as mixed populations of cells where one can identify the cells of interest by visual inspection. One can do either conventional whole cell patching or inside-out patching or detached patches. Adding to this flexibility is the ability to change the nature of the experiment on the fly. For instance, if one has a particularly good cell, one may elect to do additional biophysics or pharmacological characterization. It is unlikely that patch automation will ever rival this overall flexibility. The manual technique, however, does require a significant amount of art to master and make productive. Even the best of patchers can usually only do a dozen or so cells in a day. This level of productivity is far too low and far too expensive to support meaningful compound screening and medicinal chemistry activities. Hence, about a decade ago, a number of companies began to explore ways of automating the patch clamp experiment. For a bit of history, in the 1940s, Kenneth Cole and colleagues developed the modern voltage clamp technique. This made possible the seminal work of Hodgkin and Huxley using the squid giant axon to define the events of an action potential in the 1950s. As well, the work of Hans Ussing was taking place during this time period. During the latter part of the 50s, Leo Sternbach, who was a chemist working for Hoffmann La Roche, discovered the forerunner to Valium. The 70s, besides the decade's contribution to music and fashion, was a particularly exciting decade for those of us interested in ion channels and the tools used to study them. First, Singer and Nicholson bring together key work from the 60s to articulate the fluid mosaic model for the plasma membrane of the cell, making possible our modern notion of the ion channel as a protein complex floating in a lipid bilayer. Meanwhile, over in Russia, Kristall and Podyplachiko were developing the forerunner of the planar patch clamp technique. Later in the 70s, Sockman and Nair demonstrate that it is possible to record single-channel opening and closing events. And ion channels remain one of the few protein classes that can be studied at the single molecule level in their active state. Also in the 70s, it was found that the Valium binding site was part of the GABA receptor. So nearly 20 years after the discovery of the benzodiazepines, their mechanism of action was beginning to be elucidated. Fast forwarding past the Yuppie and Gen X decades, we arrive at the 2000s, which saw the commercialization of the first automated patch clamp platforms in 2002. To be sure, these platforms have greatly increased the interest of pharmaceutical companies for identifying ion channels as practicable targets for drug discovery initiatives by lowering the cost and, equally important, improving turnaround times for compound screening and profiling activities. One of the major drawbacks to these systems has been the high cost of adopting the hardware, as well as the high expense of maintaining these types of systems. These barriers to entry have resulted in very few academic labs or core facilities adopting automated patch clamp technology. At Fluxion, we have achieved a major generational change in automated electrophysiology by employing microfluidics, our lateral trapping technology, and an overall modular system design, which has allowed us to produce an instrument with much lower initial hardware costs, as well as lower ongoing maintenance costs, while maintaining the key objective in automating the patch clamp process: increased productivity and ease of use. Given the small footprint of the system, it easily fits into labs where space is at a premium. The systems have only 2 moving parts, so they're much more reliable and easier to work with than alternative instruments commercially available. Moreover, an IonFlux 16 can be upgraded to an IonFlux HT, allowing one to preserve the value of their capital investment in automated electrophysiology when even higher throughput is needed. So how do the IonFlux systems perform the patch clamp experiment? The next few slides will cover the principles of operation. First, we employ ensemble recording, which increases the success rate and reduces variability. Shown is what we refer to as a trap zone. The trap zone consists of 20 closely spaced microchannels in parallel. During the trapping process, 20 cells will be drawn to these microchannels and then taken to conventional whole cell recording conditions using pressure. The inside of the trap contains the internal recording solution, and the main channel with the cells contains the external solution. The entire ensemble is under voltage clamp conditions. There is a single measurement of ion flow across this collection of cells. The solutions that will flow past the ensemble are connected via microfluidic channels to the main channel. The top of the plate where the solutions will be loaded either manually or by a liquid dispensing robot has a standard SBS format, a 96-well format for the IonFlux 16, or a 384-well format for the IonFlux HT. Using a standard SBS format makes it easy for the system to be integrated with standard lab robotics for plate handling and liquid dispensing tasks. Shown on the left is an IonFlux plate, in this case having a 384-well format, ready to be loaded into the IonFlux plate reader. Bonded to the bottom of this standard plate is a proprietary polymer material containing the microfluidic channels. Then shown on the right is a close-up view of the microchannels, which have been loaded with a die for visualization in this example. We refer to the collection of chambers and their channels for a set of recording trap zones as an experimental pattern. So shown here is an alternate view of the bottom of a 384-well plate, highlighting the 32 experimental patterns. Each of the experimental patterns is highlighted with orange dye. A plate for the IonFlux 16 would have 8 such patterns. Below is a blown-up schematic of one of the experimental patterns, showing the plumbing connections and the relationship to the wells above. Taking a closer look at the experimental pattern, we see that an experiment will utilize 12 wells, which would be 1/8 of an IonFlux 16 plate, or 1/32 of an IonFlux HT plate. 2 of the wells, noted with T1 and T2, will be loaded with internal buffer. The T in this case stands for trap zone. So each pattern has 2 trap zones, an N of 2 for each experiment, if you will. There is a single in well, which will contain the cells and the extracellular buffer. The connection of the in well to the 2 trap zones constitutes the main flow channel. There are then 8 wells, denoted C1 to C8, that will contain the extracellular buffer along with the compounds to be assayed, although these could just as easily be solutions with different ionic compositions or other such variations on this theme. As well, one could load a pattern with different internal solutions in T1 and T2 to compare the effect of different internal components. For each pattern, there's a single out well into which all the solutions will flow. All of these solutions, including the cell suspension, would be loaded into the plate either manually or by a liquid dispensing robot prior to loading the plate into the IonFlux reader. Once a plate has been loaded with solutions, it is drawn into the IonFlux reader and a manifold with the recording electrodes and the pneumatic control lines is mated to the plate, shown schematically on this slide. The control of the pneumatic pressure is defined in the IonFlux control software and allows for fast control and for the compounds to be applied in any order desired. Since the fluid flow is all driven by pneumatics, there is no need for robotic liquid handling at this point, making the reader a much simpler and easier to service device. The silver chloride electrodes are connected to the amplifiers. Since both the IonFlux 16 and IonFlux HT are using standard SBS format plates, the only difference between the two platforms is the configuration of the manifold and the number of amplifiers, thus allowing for an easy upgrade path when higher throughput is needed. So the process of performing an experiment consists of loading the plate with solutions and then taking this plate and loading it into the reader. The manifold then mates to the plate, bringing the electrodes into solution and making the pneumatic control of flow possible. The pneumatic interface enables highly parallel experiments that run across the full plate at one time. Now, this cannot be achieved on automated systems that use a robotic fluid handler. Now, beyond the simplicity, one of the nice features of this design is that the bottom of the plate is completely accessible during the recording process. Having this access allows us to bring a heating element into contact with the bottom of the plate for performing experiments at physiological temperature. The IonFlux systems are the only commercially available automated patch clamp systems which allow for this type of access. We are raising the bar for performance while dramatically lowering the cost of access. Taking a closer look at the perfusion of the cells being recorded. Shown schematically are two trap zones of an experimental pattern along with the main flow channel. Get my highlighter. So these would be the two trap zones and the main flow channel. Off to the left are the 8 incoming compound lines highlighted with different colors. Solutions are prevented from leaking into the main flow channel by maintaining some back pressure as well as the continuous flow in the main channel. When a line is pressurized, in this case the green line becomes pressurized, the solution flows past the recording ensemble. In this case, in this example, activating a GABA receptor and giving rise to an inward current. Once the flow is stopped, the continual flow in the main channel clears away the ligand, bringing a cessation to the activation. So then we're ready for the next solution, and so on through the process. So that things could be added with an intervening wash step or no intervening wash step between the solutions being added. And they can be applied in any order in the system. Now, as noted earlier, the wells of the plate may be loaded manually. Alternatively, they can be loaded with a liquid handling robot. Shown in this slide is an IonFlux HT being used with a TCAN Freedom Evo liquid handling system as implemented in the Fluxion test lab. Any liquid handling robot, such as those from Hamilton, Thermo, or Aurora, could be used with the IonFlux reader, and up to 4 readers could be configured to operate with these types of liquid handling robots for those very high throughput applications. In this configuration, the reader operates as a slave device, and once the liquid handling robot loads a plate into the reader, the experimental process is then initiated and the IonFlux takes over execution of the experiment as defined in the system software. Note that while one plate is running, the next plate to be read can be loaded with solution, allowing for a very efficient and productive workflow. The system software contains all of the features necessary for defining the experiment to be executed. Shown here is the first tab of the experimental dialog, and this is where one will define the contents of the compound wells. Within the experimental sequence tab, one would be defining the pressures that will be used during the priming process, the pressures that will be used during the trapping process where the cells are being brought to the ensemble, and then the pressures that will be used to take the cells into the whole cell recording condition. And then finally, the business end of the experiment, the voltage protocols that will be applied, and the sequence of compound addition events. Highlighted is the compound addition editor, showing in this instance 4 compounds that will be added in rapid succession, followed by a flushing period where it is simply the flow through the main channel. And then this compound sequence would be repeated 2 more times. Note that the application sequence will be the same for all of the experimental patterns. However, each pattern may have a different compounds. So while the experiment is running, one can view the progress of the experiment. Shown on the right are step responses of the GABA receptor to increasing concentrations of GABA. In this case, the cells are continuously held at a fixed potential, and as the channels open, an inward current of increasing amplitude is seen as the concentration of GABA is applied for 2 seconds. Due to the pneumatic control and microfluidic channel design, the IonFlux platforms are the only automated platforms that can do this kind of experiment. On systems which use a dispensing robot, the robot must return to the compound well for the next concentration to be applied and then return to the recording chamber to add the compound. This cycle cannot be achieved within 2 seconds, and certainly not across an array of recording chambers. During the course of the sweeps, measurements are being made at the points shown with the green and yellow cursors. And the difference current is being plotted on the left window. The difference current measurement has this sawtooth appearance due to the fact that it is capturing the peak current at the yellow cursor during the sweep. Then, in the intervening sweep, the cells are once again seeing the extracellular buffer without GABA, so their current returns to near 0, followed by the next application. Now you were saying, but there were only 3 sets of compound application defined, but there are more than 3 teeth in this sawtooth pattern. This is another unique feature of the IonFlux platform. The experiment can be suspended and reinitiated during the run. As with the manual patch clamp experiment, this is particularly useful when one has a particularly good run and the decision is made that it would be nice to get some additional information from the experiment. While it would be difficult to change out the solution in the compound wells, it is possible to change the sequence of additions or add additional biophysics, such as an IV plot, to the experimental procedure. There are a number of other features, such as the amplifier control and temperature control, that we don't have time to highlight in this section. The key IonFlux specifications are that we are using an ensemble recording such that each measurement is from a collection of 20 cells. We are using loose patch technology where the average seal resistance for a given cell is in the neighborhood of 200 megaohm, although higher seal resistances have been obtained. We have very fast compound on times due to our microfluidic design, as well as very good washout times. Now, throughput rates vary depending on the specifics of the experiment. But if one had a 15-minute long experiment, it would be possible to obtain more than 250 data points with an IonFlux 16 platform, and of course, even higher throughput with an IonFlux HT platform. The amplifiers used contain all the conventional circuitry for series resistance and leak correction, as well as correction of the fast and slow capacitive components. Additional platform specifications are as follows. There are two versions, the IonFlux 16 and the IonFlux HT. The systems can be run using manual loading of the plates, but they're also compatible with commercially available liquid handling robots. The instruments have a plate reader format. The IonFlux 16 has 16 amplifiers, and the IonFlux HT has 64 amplifiers. Plate temperature control is available, which allows for running experiments at physiological temperature. The systems come with integrated control and analysis software. The IonFlux 16 has the lowest hardware cost of any automated system capable of doing more than one recording at a time. As well, the IonFlux 16 can be upgraded to an IonFlux HT. Moving on to some of the applications of the IonFlux technology and validation of the system. This is a partial list containing a number of channels that we've worked with at Fluxion. The recording column shows that we've been able to measure characteristic currents from these different ligand-gated and voltage-gated channels. The pharmacology column are channels where we've done enough work with the test system to produce a pharmacological characterization which has been consistent with expectations from literature. And finally, this optimized column are test systems where we've been able to achieve success rates in excess of 80%. It's important to note that some of these systems, like the trip channels, we've had very little time working with these cell lines. And for the most part, our activities have been driven by customer interest, and we're continually updating this collection of validation data. So shown on this slide are some biophysical properties of the Hurg potassium channel as measured on the IonFlux platform. Now, the Hurg potassium channel is a channel of particular interest to the pharmaceutical industry, since this channel has been identified as the primary target involved in drug-induced sudden cardiac death, leading to the market withdrawal of drugs like Seldane and Propulsid. Measurement of Hurg activity in the presence of compounds in development is the only bit of in vitro data required for a new drug application being submitted to the FDA. Now, importantly from this data, since we're using an ensemble and loose seal approach, the fact that these biophysical properties correspond well with expectation from the literature using the manual patch clamp technique indicate that the IonFlux system has proper voltage control of the ensemble. Now, equally important, since we're using microfluidic channels to deliver the compounds to the recording ensemble, is the fact that a battery of well-characterized blockers show their characteristic block when tested on an IonFlux system, even sticky compounds like terfenadine. So shown in this table are a set of reference compounds along with their log P values Now, the log P value is a measure of the octanol water partition coefficient, and it's an indication of a compound's stickiness. The higher the value, the stickier the compound. Then we have the IC50 values as measured on the IonFlux, as well as literature IC50 values. And note for sticky compounds, in particular compounds like pepridil and terfenadine, the literature values span a very large range. An important ability, an important ability of the IonFlux instruments is to rapidly add and remove compounds to cell ensembles across the entire plate while the current is being recorded. In a sense, the system is akin to incorporating an 8-channel rapid perfusion system for each recording ensemble. And we've looked at the GABA receptor as an example of a ligand-gated ion channel. And here we're showing 3 different GABA agonists, which correspond well to the expectation from the literature. Shown up here would be for isoquivacane, the escalating dose response, and plotted as the concentration response curves, and part of the slide disappeared. Now, of course, these would be activation curves, or EC50s, which is not what's of most interest for GABA. And despite having a 50-year history in the pharmacopoeia, GABA modulators remain a vital research interest in the pharmaceutical industry. As well, research labs continue to elucidate the functional roles of the various GABA receptor subtypes. So an understanding of their differential pharmacology will be important for the next generation of GABA modulators. And here we're showing the results of examining some well-characterized blockers and, importantly, positive allosteric modulators. And the values obtained with the IonFlux system are in reasonable accord with the expectation from literature. Now, particularly important when one is doing screening-like activities and profiling activities, consistency of the experiment is very important. When one wants to compare investigational compounds over time, so shown here is a scatter plot of current amplitudes in response to a 10 micromolar GABA stimulus for each ensemble on an IonFlux 16 plate. So there's 16 red dots here. And it's being compared to control applications of buffer without GABA here. So the bar graph is showing the average current amplitude for the 16 ensembles for a given plate, and the 8 plates being run over the course of a working day. Also shown are the Z prime calculations for each plate, shown with the red dots. So this is a very robust assay when performed on the IonFlux platform. And it is this consistency that means that one can have confidence in the responses measured early in the day with respect to those experiments done late in the day. In this experiment with the nicotinic acetylcholine receptor, alpha-1 subtype, each concentration of acetylcholine is being applied twice with interleaved control concentration also applied twice. So shown in this inset would be the, from a single recording, are the interleaved control applications showing a very consistent response throughout the duration of the experiment. So these measurements would be here, interleaved in between the escalating concentrations, which then give rise to have confidence in our concentration response curve. And also with the nicotinic receptor, as we escalate the concentrations, we see the characteristic increase in the rate of opening of the nicotinic channels. The other thing from this slide, so this experiment was done on a single HT plate, which has 32 ensembles, so there's 64 recordings on the plate. The data being shown here, compiled here with the error bars are from 62 ensembles. So 62 successes out of the 64 attempts, meaning we had a success rate of 97%. And this concentration response curve, again, agrees well with the expectation. So most important for the nicotinic experiment, we have very fast recovery times due to our microfluidic channel design. So shown in this upper panel is a trace in which the second application of acetylcholine follows 1 second after the first application. And what you can see is the second application is not producing a full response due to the fact that the channels haven't recovered. So now the same thing being done, but with a 10-second interval between the first and second pulse, we see that we get full recovery of the acetylcholine response. Now, one of the things that's truly impressive about this experiment is that we weren't adding acetylcholinesterase into the buffer that's flowing in between, which is a technique that's required on the other automated patch clamp platforms that exist. And as well, people often do it in the manual experiment. So here, just due to our efficient and continual flow, we're able to get complete wash out of the acetylcholine and recovery of the response in a very short time frame. So plotted is this recovery time to get full response. And so this is another example of how we're raising the bar in performance for automated patch clamp systems. The voltage-dependent sodium channels, or NAVs, are fast voltage-gated channels, and we use them to demonstrate our ability to record fast voltage responses. So shown in panel A are the characteristic current voltage plots for two members of the NAV family, NAV 1.7 and NAV 1.8. And these are characteristic IV plots for these two channels. In panel B, then, we have a lidocaine concentration response experiment for NAV 1.7. So we're measuring the NAV current, which is a negative value, and then as we block it, we approach 0, and then there's complete wash out of the effect. Here's a similar experiment for NAV 1.8. The experiment was set up slightly differently in that we had a wash between each application of the lidocaine. So in the concentration response curves are plotted in panel D here, and this potency difference that's seen for tonic inhibition matches well with the literature expectation. So and then a final example showing the quality of the voltage control using our ensemble approach is KB 2.1. So shown in this upper panel is the IV plot, and then the inset is conventional data taken at the Fluxion test lab. And this agrees well with the expectation from the literature, and then as well as the concentration response for a blocker TEA chloride in this case, which also agrees well with literature. So we have quality voltage control over these ensemble of 20 cells. To start summing up then, the IonFlux systems represent the next generation in the evolution of the automated patch clamp technique. At Fluxion, we are raising the bar for performance while lowering the cost of access to this technology. In terms of raising the bar for performance, our microfluidic design allows for continuous flow during the experiment and exceptional fluid exchange times. We are able to incorporate temperature control, allowing for automated experiments that can be run at physiological temperature. In terms of lowering the price, the IonFlux 16 system sells for a price comparable to a manual patch clamp rig and is the lowest cost automated platform that can make more than one measurement. The IonFlux 16 systems are validated automated patch clamp platforms which will help to accelerate your research by minimizing the time needed for training, increasing your data acquisition capacity, and improving the consistency of results. In conclusion, the IonFlux systems brings automated patch clamp technology within the financial reach of the academic researcher. Additional product information and application notes are available at our website, www.fluxionbio.com. As well, if you'd like additional information or system quote, contact sales@fluxionbio.com or your local representative. I'd like to thank you for taking time today to learn about the IonFlux system. And I would like to encourage you to contact us to see if an IonFlux system may be right for your application needs.